Vulnerability to reservoir reseeding due to high immune activation after allogenic hematopoietic stem cell transplantation in individuals with HIV-1, a study performed by the ICISTEM consortium funded by AMFAR. Worldwide, there are over 37 million people living with HIV, and only 3 are considered to be cured. The first breakthrough was achieved 13 years ago when an HIV-positive blood cancer patient, the Berlin patient, received a stem cell transplant from a donor with a mutation in the HIV entry receptor. Allogenic stem cell transplantation became the first and so far only medical intervention that has reportedly cured HIV. While in most people with HIV, viral rebound is observed within few weeks if antiretroviral therapy is discontinued, in the case of the Berlin patient, the virus has remained undetectable since 2008. Since then, there were several attempts to reproduce the success. However, in most of these cases, the patients did not receive stem cells with a CCR5 mutation. After the transplantation, there was no detectable virus, but still the patients rebounded within 1 year after stopping antiretroviral therapy. At this point, it is not clear if the CCR5 mutation is essential to achieve HIV cure. However, 2 new cases of cure or durable HIV remission, the London and the Düsseldorf patient, further emphasize the importance of the CCR5 mutation. To understand the clinical, immunological, and virologic factors associated with HIV remission after stem cell transplantation, it is necessary to analyze these patients comprehensively. Since 2014, the ICISTEM consortium has followed HIV-infected individuals who receive a stem cell transplantation for other medical reasons, like for example leukemia. As preparation, the individuals receive a chemotherapy that reduces their own blood cells, including cancer cells and HIV-infected T cells, to a very small number. This leads to immunodeficiency and potential for opportunistic infections. To replace these cells, the individuals receive a stem cell transplant from another person. Stem cells have the potential to differentiate and to develop in different types of immune cells. T cells from the donor help the patient to control opportunistic infections or reactivation of chronic infections. It will take several months or years to reconstitute the immune system, and during a short period after transplantation, donor T cells and few remaining recipient T cells are present at the same time. Now donor T cells recognize recipient cells as strange and attack them, including also HIV-infected cells. This explains the immense decrease of infected cells in these individuals. However, we have observed that both donor and recipient cells receive signals to get activated and proliferate. The few activated CD4 T cells may release HIV particles, and highly activated donor cells also present perfect targets for reinfection. And if antiretroviral treatment or genetic barriers are not efficient, this may lead to reseeding of HIV in engrafted donor T cells. Donor T cells take over and eliminate the few remaining recipient cells, and among the donor cells appear cells that recognize HIV, indicating that indeed HIV products were produced by recipient infected cells during the initial reconstitution period. Months after stem cell transplantation, only donor cells can be detected. Immune activation decreases. HIV-specific cells can still be detected. Years after the transplant, a few HIV-specific CD8 T cells can still be detected, but they appear not functional enough to control reactivation of HIV if antiretroviral treatment is interrupted and some infected cells are still present. Eventually, interventions to establish genetic barriers or to reinforce immune responses may be needed to achieve HIV remission.